Hello students. Today we'll be discussing on a new topic which is on the DNA structure. So what we can expect as a question most commonly. In the exam they might ask you to explain the structure and write the features of human DNA, or they might ask you to write about the Watson-Crick model of DNA, or they might ask you to write about the B form of DNA. Okay, any of these can be questioned in the exam. They might ask you in any form. So what is the answer? Now before we go into that, what are these DNA? That is deoxyribonucleic acid. They are basically the nucleic acids. And what are nucleic acids? Nucleic acids are the polymers of nucleotides. And what are the different nucleotides? We have adenine, guanine, cytosine and thymine. Four different nucleotides. And specifically the ribose sugar, the sugar part of the DNA is in the deoxy state. Okay, that's why it is called as DNA, deoxyribose nucleic acids. And one more short answer question, they might ask you what are the different types of DNA. There are majorly the three types of DNA: A form, B form and Z form. So these are the three different types of DNA. Coming to how to write about the DNA structure or how to explain this, you should be able to at least recognize the structure of DNA, how it is basically made up of. It is made up of a nitrogenous base, which is adenine, guanine, cytosine and thymine, a deoxy sugar and a phosphate group. So these are the three things which makes it a nucleotide. And DNA is a polynucleotide, means it is made up of many nucleotides, multiple nucleotides. And monomeric unit having the deoxyribonucleic acid, which contains deoxyribose. Why we call deoxyribose is, here instead of OH group we have H group. Instead of OH group we have only H group. The oxygen is removed. That's why the ribose sugar is called as deoxyribose. And what are the different nitrogenous bases? As I mentioned, adenine, guanine, cytosine and thymine. These are the four nitrogenous bases which are present in the DNA. Phosphate group forms the structural role, and link between the nucleotides is by the phosphodiester bond. Okay, a phosphodiester bond is formed between two nucleotides to form a long chain, or polymerization occurs. Then coming to DNA structure, the predominant DNA structure which is commonly seen is the B form of DNA, which is explained by a scientist James Watson and Francis Crick. So that's why this model of DNA is also called as Watson-Crick model of DNA. In the exam they might ask you to write Watson and Crick model of DNA or explain it. What they told, that DNA is made up of two chains, two polynucleotide chains. You can see in the figure also we have one chain and second chain, and you can see some bonds here. They are the hydrogen bonds holding these two strands to form something called as double helical structure. And do they have any sides? Yes, they have two sides, that is a starting and ending point. We call that as 5' end and 3' end. If we see here, this is one chain which has 5' end, and the end of this is 3'. Similarly, other prime 3' and 5'. So they are double helical structure containing the sides 3' end and 5' end, and the two chains are coiled around itself with a common axis, a central axis. Okay, so these two are they parallel? No. If you see here, this is starting at 5' and ends here 3'. Other chain starts as 3' and says 5'. If you look from up to down, that is top to bottom, if we see, they are running anti-parallel. So both the chains are anti-parallel and they are right-handed double helical structure. What are the bases they have? They have purines, they have pyrimidines. Purines include adenine and guanine. Pyrimidines include cytosine and thymine. And these both are present at the core. If you see this, they are the base pairing between the structures. And these contains a minor grooves and major grooves. Okay, the DNA structure is converted or read as major groove and minor groove. And each rotation contains 10 base pairs. When it makes a one rotation from here to here, it contains 10 base pairs in between. 10 base pairs. And what is the distance? It is the 3.4 nanometer total. So when it contains 10, 3.4 divided by 10 will come to 0.34 nanometers. Between what? Between each nucleotides. A distance between each nucleotide is 0.34 nanometer. And how much is the diameter? It is 1.9 to 2.0 nanometer is the total width of the helical structure. So these points you have to mention by drawing the structure of DNA. Next, the sugars and the phosphate forms the backbone. Okay, if you see, the backbone is formed by the phosphodiester bond, which is bonding the sugar molecule and the phosphate molecule, and they are hydrophilic in nature. So that's why they are present on the outer surface. When it comes to the bases, that is the purines and pyrimidines, they are hydrophobic. So they occupy the inner structure. Okay, they are present inside the helical structure. And what is the pairing? That is the pairing is always adenine binds with thymine and guanine binds with the cytosine. And how many bonds? Adenine double bond with thymine, guanine triple bond with the cytosine. So by the bonding also you can see G pairing with the C is the strongest bond. Okay, it is the strongest because it has triple bond when compared to the double bond. Okay, so this is having some rules. This rule can be a short answer question in the exam most commonly. That is the Cherkov's rule of base pairing, which is where the amount of adenine, that is amount of adenine is always equal to amount of thymine, and amount of guanine is always equal to amount of cytosine, because they are pairing with each other. So if there are 10 adenine, there will be 10 thymine. If there are 10 guanine, there will be 10 cytosine. If you see, these are purines. So purines divided by pyrimidines is always equal to 1. So what is the second law is telling? The amount of purines and amount of pyrimidines, when you make the ratio, it is always equal to 1. It's always equal to 1. Okay, so with this we'll complete our question, which is explaining the structure and features of human DNA, which we have seen that there are two strands, both are anti-parallel, they are having directions, that is 3' and 5' ends. There is a phosphodiester bond which is keeping as a backbone of the DNA structure. The sugar moiety and the phosphate are hydrophilic in nature, so they are present at the peripheries. The adenine, guanine, thymine, cytosine, which are purines and pyrimidines, are hydrophobic in nature. They are present in the core. Adenine always binds with thymine with double bond, and guanine binds with the cytosine with a triple bond. So the pairing between guanine and cytosine will be the strongest. Okay, so these are the features, and also you have to mention about the Cherkov's rule. Okay, don't forget about the Cherkov's rule, which can be a short answer question. So with this, we'll conclude on question explaining the structure and writing the features of the human DNA. Thank you very much.